Hi, everybody. It's so nice to see a packed house. I'd like to do a little shout out to Professor Passarelli's Saddleback College class that came here all the way from Mission Viejo. That's a long drive on a Friday night. So we welcome you guys. Thank you guys for making the drive. It's fantastic to have such a full house. Before I introduce tonight's speaker, I'd like to talk a little bit about our speakers lined up for next year. We have them almost all lined up, but before I tell you all the speakers, I want you guys to know how grateful I am for a certain person that's been helping us do this lecture and get this series going. And someone that I've been organizing this with since it started, and that's Mike Schott. And this is going to be his last one. He didn't mention it. I'm going to mention it, and you can get mad at me later, but he's retiring in January. And so this is his last lecture that we're doing as a team. And congratulations on your retirement, Mike. We're going to miss you. Please attend the rest of the lectures. We're going to miss you. So congratulations.
This might be not the last time you see me.
All right. Thank you. So we have 5 of the 6 speakers lined up for next year. Some of them have been rolling through the slideshow, but let me just go through them very briefly. On February, they're also all on our website. February 2nd is Dr. Kevin Lafferty from the U.S. Geological Survey at UC Santa Barbara. He'll be talking about parasites and food webs. On April 6th, will be Dr. Karen Martin from Pepperdine University, and you may have guessed she'll be talking about grunion, surf, sand, and silver sides, research and outreach with California grunion. On June 1st, it will be Dr. Christine Woodcraft from California State University, Long Beach, talking about designing solutions to restore wetlands in a time of change. The August 3rd speaker is who we're still working on October 5th will be Dr. Craig Stanford from USC, and he'll be talking about biodiversity work with turtles and tortoises. December 7th, Dr. Ian Fenty from NASA Jet Propulsion Laboratory, and he will be talking about warming oceans, melting ice, and rising seas. So I hope to see you guys at all of those. But tonight, it is my pleasure and honor to introduce tonight's speaker, Dr. John Dabiri. Dr. Dabiri is a full professor of civil and environmental engineering and of mechanical engineering at Stanford University. He earned his bachelor's degree from Princeton University and his master's and PhD from Caltech. His research focuses on science and technology at the intersection of fluid mechanics, energy and environment, and biology. Honors for his work include a MacArthur Fellowship, an Office of Naval Research Young Investigator Award, and a Presidential Early Career Award for Scientists and Engineers. Popular Science magazine named him one of its brilliant 10 scientists for his research in bio-inspired propulsion. Bloomberg Business Week magazine listed him among its technical innovators, and MIT Technology Review magazine named him one of its 35 innovators under the age of 35. In 2014, he was elected a Fellow of the American Physical Society. The title of tonight's talk is, Do Swimming Animals Mix the Ocean? Please help me welcome Dr. John Dabiri.
Thank you. Thanks for having me. It's really a pleasure to be back here. This is in many ways where a great part of my career got started. When I was at Caltech as a grad student, we started becoming interested in jellyfish and their biomechanics and what we as engineers might learn from them. And so, of course, the first step was to try to find jellyfish. And so I Googled jellyfish, and you'll find a few aquaria around who have the jellies, but mostly when you call them and ask if they can help you set them up and study them, they say, no, it's too difficult, we don't have the resources. But then I got in contact with Mike Schott, and he actually wrote me back, and he said, it's probably going to be difficult for you to do this at Caltech, but why don't you come over here to the Cabrillo Aquarium? And so for the following 13 or 14 years now, we've had a continuous interaction with the aquarium, It spawned dozens of projects, literally involving undergraduates, graduate students, high school students, other faculty colleagues of mine as well. And so this place is really a special place for me to come back to give this lecture, and I'm really appreciative of all of you for your support of the aquarium and its mission. So today, I thought I would tell you about an avenue of our research that actually dates back to those very early days of our research, and I've titled it, "Do Swimming Animals Mix the Ocean?" In part because it's a question that at first you might say, of course not, that animals are too small, the ocean is a huge place. And so it's an unexpected question to ask, but as hopefully you'll see today, there's some surprising answers in store for us. Now, for me, this work began with the gentleman behind me, Walter Munk. Some of you will know him. He's a professor at Scripps in oceanography there. And when I first started getting interested in ocean science, I read one of his classic papers. It's called Abyssal Recipes, dating back now more than 50 years ago. And as I was reading this paper, which talks about how the ocean works, it lays the foundations for what we know today, I noted this particular sentence or paragraph where he notes, "The most abundant organisms in the abyssal sea," so the deep sea, copepods, mysids, euphausids, these are krill basically, squid, and the nectonic bristlemouths, are all engaged in diurnal vertical migration over a vertical distance of over 1 kilometer. So all of these organisms on a daily basis are making these large vertical migrations in the ocean. And he notes that this migration is associated with some transport of ocean water. And so to me, this seemed like a really interesting and sort of provocative idea that as these animals are moving, that transport of water might actually be something significant. And so it begs the question, do swimming animals mix the ocean? But I'm going to talk about this from two different perspectives. So the first is going to be, when I say mix the ocean, we're going to talk about contributing significantly to maintenance of the global ocean circulation. So the way that our Earth works, the reason that we don't all eventually freeze and die is because the heat from the sun is transported by the ocean from the equator to the poles. And some of that water then sinks down to depths, and so you can see it illustrated here in this cartoon. And the mixing of denser deep water into lighter surface water requires some input of energy. And the speculation is that perhaps these animals, as they're swimming vertically in the ocean, might be contributing to this great ocean conveyor belt, as it's sometimes called. So that's one context in which this question is asked. The other is in the context of transporting carbon. So as you may know, our anthropogenic generation of carbon dioxide is a big problem right now in terms of global climate change. The ocean is actually the primary sink for the carbon that we emit. In fact, half of our carbon emissions since the Industrial Revolution, they end up in the ocean. Now, what we know today, this is from a paper about 15 years ago, 17 years ago now, is that there are biological processes where CO2 that's in the atmosphere ends up in the ocean through phytoplankton. These are small organisms that undergo photosynthesis. And then they might be consumed by larger organisms. And eventually, that carbon gets sequestered into the deep ocean, as is illustrated here. Now, there are also upwelling phenomena, so just vertical currents in the ocean you can think of, that also can lead to vertical exchange back up into the atmosphere. And so it's important that we understand what happens in this physical process, because if we want to predict the fate of our world in terms of climate change, this is going to be one of the important variables we have to get correct. Now, one of the challenges is we know a lot about the ocean surface from satellite images, but if you go down 10 or 20 feet below the surface, we really know very little about our ocean. Less than 5% of the ocean by volume has been sampled to date. And so the question we ask here is, as the animals are swimming in the ocean, are they impacting this process? Because if they are, that has huge effects on things like climate change and so on. So that's another context. Now, what do we know today? So if you ask a card-carrying oceanographer, they'll tell you, first of all, one of the things that's important for maintaining the ocean is the wind. The wind blows along the surface of the ocean, and it creates eventually surface waves, but also what we call internal waves. It's similar to the waves that you can see visibly. They're just occurring below the ocean surface. And it's in green here, so it doesn't show up so great, but the energy input here is 0.8 TW is trillion watts. So it's a huge amount of power that's being put into the ocean by the wind. The other number that I want us to keep in mind is what we call turbulent dissipation. Just think of this as another measure of how much energy is being consumed in this process. And the way that we sometimes refer to this is in terms of watts of power per kilogram of water. And it's a small amount when you average per kilogram of water because there are trillions of kilograms of water in the ocean. So the number to keep in the back of your mind, 10 to the minus 5, so that's 0.00001 watts per kilogram, 10 to the minus 5 watts per kilogram. Keep that number in the back of your mind because we'll come back to it later. So the other important phenomena that oceanographers know about is what happens at the bottom of the ocean. So tidal currents occur due to the position of the moon, and those tidal currents, as they run over topography, the bumpiness at the bottom of the seafloor, that also creates those same types of internal waves that again can lead to churning and mixing of the ocean. And the thing to note here is it's the same sort of amount of energy here, 600 to 900 billion watts of power, almost a trillion watts of power, and that 10 to the minus 5 watts per kilogram. Now, I show you this just to note that today what we know, for those on this side, it says 2 trillion watts in total are required to maintain the ocean circulation. So we know we need 2 trillion watts of power to maintain the circulation. These numbers are rough estimates. They get you around 2 trillion if you add up those two sets of numbers. And so most oceanographers will say, well, close enough. We probably understand how the ocean works. It's a combination of wind at the surface and tidal currents that's generating the energy that mixes the ocean. So then there's this question of animal swimming that Walter Munk mentioned. And so we can ask, could that also be playing a role in the maintenance of ocean circulation? Well, here's what we know about the animals in the ocean. This is a sketch based on net tows. So you can imagine taking a ship out to sea, dropping a net down, pulling it up at different depths, and figuring out what's the biomass at different depths in the ocean. We can also use acoustic measurements. So you send out sound waves, they'll scatter off of the biology that's there, and at a given depth, we can figure out the populations. So what we know from this is that in the epipelagic, as it's called, very ocean surface here, and the mesopelagic just below that, is where we think most of the biomass is, at least the biomass that we can categorize by those two methods, net tows and by acoustic measurements. And so these are the things that you typically think about. Now, at the very bottom of the ocean, the abyssal plain here, so you're talking going down 4,000 meters in some cases or deeper, we know there are organisms there as well. Now, the exciting thing in recent years, only in the past decade, have we started to have new technologies that allow us to put cameras in the middle ocean here, this middle region here, where this scientist has drawn a very skinny bar indicating the assumption that there's not a lot of biomass. In fact, I don't think that's going to turn out to be correct. This is just from last year in the Mariana Trench, a jellyfish here with this beautiful gold bell at 3,700 meters. So the problem is, if I put a net down to that depth, if I even could do it, this thing is going to be a bunch of goop by the time I pull it up. So there's not a real good way to take a census of these gelatinous organisms. And we also don't have the technology to be able to resolve this length scale. This is maybe a centimeter or two at that depth using acoustic measurements. So this is just an aside, and Mike referred to this earlier today. We still know exceedingly little about the ocean and its biomass, and so we're going to need to fill in this picture better to get at this question, but I digress. The thing I want to note here is that Bill Dewar's group at Florida State did some estimates of how much mechanical energy might be available in all the biomass in the ocean. And the way they did that was by making the logical argument, we know that everything in the ocean gets its energy from the surface, so let's just do a counting of how much energy goes in through photosynthesis. And so that net primary production, as it's called, gets you an estimate of about 1 trillion watts, which is on par with the winds that we saw in the previous slide, with the tidal currents that we saw in the previous slide. So in principle, there is enough energy there for this to be a significant effect. Now, the first recent sort of work, recent meaning after that 1966 paper, was several decades later. A group looked at data from 100 different aquatic species, varying from krill to the sperm whale here. So huge ranges from millimeters to tens of meters in length, going out to 1,000 kilograms in mass. And what they wanted to understand is, what is the energy dissipation that those organisms might make? Remember on that previous slide, there was that weird number 10 to the minus 5 watts per kilogram that we said was the amount of energy being dissipated by the winds and the tides. So this is sort of an eye chart here. I just want to point out this last column here. What they did was look up a bunch of different species and then make models based on their mass, their abundances in the ocean, how fast they swim, what's their swimming efficiency, so how much energy do they put in the water when they swim. The take-home message here is that all of these numbers here, we don't care too much about this left-hand side. The right-hand side is that the order of magnitude was 10 to the minus 5 watts per kilogram. So they estimated for all of these animals that the amount of energy that they're dissipating is very similar to what we see from the winds and the tides. Again, another piece of evidence suggesting that these animals might be important for ocean circulation. And then the kicker was probably Eric Kunz's group. This is a group from British Columbia in 2006. Now they went out and at sunset, when they knew a large group of these krill would be vertically migrating, they measured how much energy was being dissipated in the process. So this is actually the sort of smoking gun you'd want to see, measuring the mixing that's occurring as these animals are migrating. And this is a very difficult piece of data to get, because if I go out off the coast here or up in Monterey, closer to where we are at Stanford, it's hard to know exactly where to sit and look for a vertical migration. So they got this data by accident, basically, but that's how you get published in Science sometimes. So this paper is from Science. It shows this migration here. So this dark color is this zooplankton layer, as they call it. So these are what they think are the krill. This is 7:30 p.m. and going later into the evening. And you'll notice this layer, which this comes from that acoustic method, sending out the sound waves, it's scattering off the layer. You'll see it's rising vertically. So this is depth here from about maybe 90 meters to 40 meters. And as that group is migrating, they're measuring how much energy is dissipated. And the only thing I want you to take from this is again, depth versus time. Anywhere that you see these red spikes here in their record of this is the measurement of the turbulence, they actually saw dissipation rates 10 times higher. So for those who are sort of rusty on the math, 10 to the minus 4 is actually bigger than 10 to the minus 5 by a factor of 10. So they saw 10 times more energy being dissipated than even the previous authors in their study had predicted. So all of the data to this point seems to suggest that animals are having a significant impact on the ocean. However, the same group tried to reproduce this result, and so this is from a master's student, Shani Rousseau. She was asked to follow up that initial data for her graduate work. And this is one of the sentences that they note. First of all, I guess I'll just say in my own words that in subsequent field measurements, they had a hard time reproducing that earlier result. Again, part of the issue is you don't know where to go in the ocean to find one of these migrations. They found a couple that seemed like they would be similar migrations, but they conclude that based on their sampling to date, this turbulence production is not efficient. Basically, they just didn't see it happening often enough to believe that those previous results were representative of what happens in the ocean. Now, at this same period of time, and the narrative I'm telling you here is very much what occurs in many fields of science, where there's a debate that swings back and forth. And so there was a lot of momentum for this idea of biomixing. Then Eric's and other groups had a hard time observing it. They said, okay, maybe this is not a real thing. And then Andre Visser in Denmark published a sort of rebuttal paper where he noted that really what we've been measuring so far is just this churning of the water. That's not really the same thing as mixing. So to mix something, think of your coffee in the morning. I have my coffee, I put the creamer in, and I stir it. So in order to mix something, I have to have two different things, cream and coffee, for example. In the ocean, I need to have water that has different properties, meaning a less dense amount of water and a heavier water. So those different types of water are going to be separated by really large distances. So the less dense water, the lighter water is going to be, say, at 50 meters. The heavier water might be at 100 meters. And his argument was that what you really want to know is how much of the energy is actually contributing to mixing over those really large length scales before the energy just gets dissipated as heat by viscosity, and we don't need to go into that. The point is this. What he shows is what you actually care about is this length scale. He calls it a buoyancy length scale B, And how big is that? That buoyancy length scale being how far apart are these different parcels of water compared to the animals. His argument is that the animals themselves are just too tiny to matter. So an animal that's a centimeter in size, he argues, is just going to mix that local water. And so all that water within a centimeter of itself is not that much different from the rest of it. And so he says because the animals are going to sit in this range here, sorry if the light here is a little bright, maybe if we can turn that down a bit. Maybe I can do it here. Let's see. Hopefully I don't hit the self-destruct button. I think that might have gone down a little bit. Because the animals are sitting in this region here, the mixing efficiency that he predicts here is less than 1%. So the conclusion he makes is basically the animals themselves are too small to make a difference. And so this is 2007. I've been working on this problem for about 4 years. I had read that paper by Walter Munk that got me really excited about this idea. And so I figured, okay, at this point, maybe I just need to ask Walter. So I emailed him. And you can see here my subject line, Ocean Mixing by Swimming Animals. This is 10 years ago now. And Walter wrote me back. In fact, I think the same day he wrote me back. And he says this, "Dear John, it was partly an attempt at humor when I suggested many years ago that diurnal migration could lead to appreciable mixing. And I was amazed at recent papers who think this is not a joke." So, not the most encouraging thing to hear after 4 years of working on something. But he goes on, fortunately. He says, and people thought it was a lunatic idea when Carl Wunsch, she's an oceanographer at MIT, and I suggested that the moon via lunar tides could have anything to do with mixing, and now that's generally accepted. So that picture that I showed you on that earlier slide showing the tidal currents, that's an established part of our understanding of the ocean, but when he first proposed it, people thought they were crazy. And he says here, "I must prefer the era when we were called lunatic to the era when people say we all know." And for the students in the audience, keep that in mind when your teachers try to convince you they know everything. But he ends with a really key sentence here that I've kept with me. He says, "I can only say, work on problems you most enjoy. Strange things can happen underway." And so that led us to say, okay, well, let's go ahead and see what we can figure out in this space. And so we decided our objectives for this research were going to be twofold. First, we wanted in situ, in the ocean, to try to measure this mixing efficiency. Andre Visser had made a compelling case that the animals individually are too small to make a difference to mixing over very long distances. But let's see about trying to measure that. And then again, coming from an engineering background, we wanted to know what are the physical mechanisms? What's the physics behind this process? And perhaps we can make some progress here. And so as I mentioned earlier, we had already been studying jellyfish because of our interest in bio-inspired propulsion. The animals tend to be very energy efficient in how they move, and so we thought we could learn something there. And so we had already been doing work in places like Jellyfish Lake in Palau. So this is an image there where you have these really unique habitats where these animals exist in very high abundances. And so this work was led by a wonderful student here. Unfortunately, the title here is washed out, but this is Kakani Young. She's now a principal engineer at the Monterey Bay Aquarium Research Institute. She got her master's degree in aeronautics at Caltech and her PhD in bioengineering there. And before that, she was actually on the US national figure skating team. So she's a very talented athlete as well. She does everything, basically. But for her thesis, she developed some technology that would allow us to measure the flow fields that these animals were creating as they swim, and to do it while scuba diving, so that you're right there in the middle of the animals measuring them. Before I get to that, here's just a video showing some dye, again, giving you a sense of what happens when these animals are swimming. The dye helps us to visualize their flow. So this is a Jellyfish Lake here. You'll see some dye injected here, and they create these swirling vortex rings on either side as they move. And so we thought, again, because of a relatively simple body shape, it's basically like a hemisphere. The wake structure has these vortex rings that are pretty easy to visualize. They would be a good starting point for trying to understand this. And so as a part of her PhD research, she developed a technology which we call the self-contained underwater velocimetry, so velocity measuring apparatus, or scuba, which here she is in the Caltech swimming pool with one of the devices. Here's a few later iterations. And what this device allowed us to do was to measure the flow around these animals in the ocean for the first time. And so to give you a sense of this device, I thought I would take a clip, and let me plug in the sound here, from a talk she gave, a TED Talk that she gave a couple of years ago. And so she'll tell you a little bit more about the technology.
And so a single scuba diver can go anywhere from the surface to 40 meters or 120 feet deep to measure the hydrodynamic signatures that organisms create. And now I want to, before I begin, I want to kind of immerse you into what these kinds of measurements require. So in order to work, we actually dive at night. And this is because we're trying to minimize any interactions between the laser and sunlight. And we're diving in complete darkness because we do not want to scare away the organisms we're trying to study. And then once we find the organisms we are interested in, we turn on a green laser. And this green laser is actually illuminating a sheet of fluid, and in that fluid, it's reflecting off of particles that are found everywhere in the ocean. And so as an animal swims through this laser sheet, you can see these particles are moving over time. And so a lot of you think, okay, this lady's crazy, right? Well, we actually risk our lives to get this kind of data. What you're going to see is that on the left, these are particle images that shows the displacement of fluid over time. And using that data, you can actually extract what the velocity of that fluid is. And that's indicated by the vector plots that you see in the middle. And then we can use that data to answer a variety of different questions, not only to understand the rotational sense of that fluid, which you see on the right, but also estimate something about energetics, or the kinds of forces that act on these organisms or on the fluid, and also evaluate swimming and feeding performance.
So that's, I think, a really good summary of the technology that she developed as a part of her thesis. And here's a slowed down clip of one animal that we measured over here in Spinnaker Cove. And so this was data, I don't know if Mike Schott remembers this data set, but he actually suffered a lot to get this particular data set. So he and I and Kakani would go out to Spinnaker, and because I'm not much for diving, I was sort of the deckhand, and he and she would go down. And so this was a particularly cold evening where we usually would dive at night, as she mentioned. We probably wrapped up around midnight, maybe later than that. They finally got up out of the water and dried off and everything, and it was cold, so I had my hand shoved in my pockets. And so we were ready to leave, and they said, "Okay, John, where's the keys to the truck?" And so I quickly threw my hands out of the pocket, and the keys arced in the air and splashed into the water and sunk down. And so Mike was a really good sport. So Kakani and I had come in this pickup truck. He had driven separately, and if I was him, I would just hopped in my car and said, "See you later." But he jumps in the water again. He swims down. I don't know how he found the keys in there, but he found the keys for us. So thank you, Mike. But this data becomes really valuable for us because we're able to now map. Each of these little yellow arrows is telling you at that specific location what's the speed of the water at that particular spot. And so you can see as the animal swims, you get this complicated flow pattern that, as Kakani mentioned, allows us to be able to directly measure how much energy these animals are putting in. What is the turbulent mixing that's occurring, and what's the efficiency? And so the short answer to that earlier question of answering, what's the empirical efficiency, is that right here in this green box here, 26% with some pretty big error bars was the efficiency of the mixing that we saw in these animals, as opposed to the prediction by Andre Visser that it should be 0.1%. So we were finding much higher efficiencies actually in the water measuring them than what the theory had predicted. And in fact, the efficiencies here were comparable to, these terms aren't really important for our purposes, but these are other processes that we know are important in the ocean for mixing. And so the efficiency of these animals was right in line with all of those other processes. So the question then was, okay, what's the physical mechanism going on here? Because Andre Visser and others made a very good point. These animals are relatively small. How could they efficiently mix water properties that are occurring over big distances? So what we did was to go back to some of these videos, and if you stare at these for a long time, you start to make a shift, and originally what we were really interested in is what we call wake mixing, which is that as this animal's swimming here, you'll see the dye injected, it creates these swirling eddies. But again, those swirling regions where you might imagine a little spoon there stirring the water are pretty small. On the other hand, if you look right behind the animal, you'll notice this bit of dye that's trapped behind the animal and is getting towed along with it as it swims. You'll see that again here as we release the dye. So there's another effect going on here, which we call induced drift. So a drifting of the water behind each of these animals. Here's a different way to see it. It's a bit clearer in this video here because we're zoomed out a bit more and we're going to be moving along with the animal. So here's that initial clump of dye, and you'll see this long trail behind the animal as it swims. It's dragging the water with it. One more view, this goes back to that PIV technique. Hopefully you in the back can see that there are little particles that we've seeded in the water. These are just tiny glass beads, so this is in the laboratory. But you can see that if you train your eye right behind where the animal is, you can see this mass of these particles that are just moving right along with it. Now, this is not the animal pulling itself up by its bootstraps, so to be sure on the sides here, you can see as this video loops again, there are these rearward jets. So it's creating this backward jetting flow. Newton says for every action, there's an equal and opposite reaction. So the reaction to that backward jetting here and here is that the animal propels it forward, but it's carrying water along with it as it swims. So it turns out that this mechanism is not something new. It actually, what we started calling a Darwinian mechanism, not after the Origin of Species author, so this is Charles Robert Darwin. He had a grandson who's lesser known, Charles Galton Darwin, and he wrote a paper simply titled Note on Hydrodynamics, very simplistic title, but a very important paper from the mid-1950s. And in that paper, he thinks about the problem of if I have some object in a flow, and this is going to be a simplified case of what we just saw before. So just think about a sphere. It's rising in water. And in fact, we're going to think about a case of what we call a potential flow, which is a case without any viscosity. So the molecules don't interact with each other in the more complicated way that a real fluid does. But the benefit of this potential flow, as it's called, is you can write some equations on paper to describe how every single bit of water moves during this process. And so the physics are that you have an initial forward motion of those water particles. You have some lateral looping. The important thing is that there's this permanent forward displacement that starts to kind of look like what we just saw with that dye being permanently displaced by the motion of the jellyfish. Now, okay, this is a theoretical model. It turns out this is something you can see yourself. I don't recommend this, but here you can see a cool demonstration of that same kind of looping. So I found this on YouTube. I don't know exactly what the circumstances were here. But that semi-truck is going by, and it's like that object that we just showed, and you can notice this smoke doing that looping motion that's very similar to what we just talked about. So this is a real physical process. Again, don't do this. Now, the other thing that Darwin looks at is if I connected all of those points at any given time. So imagine this was an actual surface and I tracked, it's just the forward motion. So I'm not tracking the part that wraps around, but just how much is each particle being displaced upward as this object moves. You can see it starts to trace out this horn of water here, which he calls a drift volume. And so the interesting thing about this drift volume is that you can take the drift volume and divide it by the body volume, and you get something which he calls an added mass coefficient. This is the added amount of mass that's carried by an object as it moves, and it's only shape dependent. So for a sphere, the added mass coefficient is 1/2. The amount of water that's drifting with it is just 1/2 of the mass that's displaced by the sphere itself. For a cylinder, it has a value of 1. The important thing is that this drift proportion depends on the shape, not the size of the animal. And so that became important as we were thinking about this in the ocean context, because remember, for that turbulent mixing process, there were models that suggested a small organism can only mix a small amount because of the effect that a viscosity would have. Here, there's no viscous effect. There's no viscosity yet. Now, what is the effective viscosity? So here's that potential flow video that I showed you before. The way we think about viscosity in fluid mechanics is with a parameter called the Reynolds number. You don't need to know what the Reynolds number is, except to say that for a value of 100, it means there's some viscosity in the water. Imagine the difference between taking your spoon up through water versus taking it up through honey, for example. Those would have different viscosities. For this case with real viscosity, you can see that the drift volume is actually enhanced. Let me go back a second so it'll replay. On the right-hand side there. See if I can get this to play. Well, I'll show you this picture here, which is a couple of still frames of that. So as the Reynolds number gets smaller, the viscosity effect gets larger and larger. And so this doesn't have to be in the ocean, you actually changing how much viscosity is there. But a small organism is going to feel the molecular viscosity of the water more than you or I would swimming in that same type of water. And so the effective viscosity here is to pull up much more fluid, say at a Reynolds number of 1 than at a Reynolds number of 10. And these are the added mass coefficients that we calculate. Remember, in the case of that flow with no viscosity, the coefficient was 1/2. Here you're getting a coefficient of 4. So it actually carries 4 times its own displaced volume with it as it moves. And so this viscosity enhances induced drift. And so again, that was theoretical work, but we wanted to see what actually happens in the real world. And so Jana Naroth was a grad student in the lab who finished in 2012. She also worked here at the Cabrillo Aquarium on many occasions. And so the experiment she did at much smaller scale, so again, this is Reynolds number of 10, like I said, smaller Reynolds number, larger viscous effect. She wanted to see how much drifting a small ephyra would make. So you can see these over here in the nursery, I'm sure, these little tiny organisms. What she did was to start the animal in this blob of dye and let it swim on its own. And so you'll see here, it'll come into focus, that as it swims through the water, it carries with it, and it creates this really neat, like, sort of seasonal Christmas tree kind of a shape here, as it's pulling through. So it's creating this flow effect that is much larger than its own individual body size. And as I sort of sketched here, here's the length scale of the animal, and here's the length scale over which water is transported by that tiny animal by this induced drift effect. And it's much larger than the length scale of the animal. So this gave us a sense that this is a real effect. And so we started to imagine then that in the ocean, you could imagine that vertical migration of even centimeter scale copepods or krill or other organisms rising and carrying with them this higher density water at depth. It's denser because it's colder, and sometimes it'll have a higher salt content. And bringing that into the region of that lower density water. And now what we found from some later work was that by having multiple animals together, that transport could even be further enhanced. And I'll talk about that in the back half of the talk here. But for now, the other thing I wanted to note is that what will typically happen if you take something that is more dense and you lift it above what's less dense, you'll get what are called overturns. It'll want to fall back down. And in that overturning process, then you could potentially get larger scale mixing than what you would get from any single organism. And so this was the picture that we started to think about, where the overturn volumes could scale with the area of the aggregation. In the ocean, these aggregations can be kilometers in width. They can be several hundred meters in depth in some cases. And so this was work that we featured in Nature that had one of Kakane's really nice pictures here showing that drifting effect. Now, it led to a lot of important questions. First of all, we studied this in jellyfish because they were convenient for what we had previously been doing. But you could ask, what other organisms, what would this look like for other organisms? What locations around the world would this matter? In different seasons of the ocean, the density stratification, the gradients, the changes in density can move vertically to being very close to the surface or being very deep. So what seasons would matter? Which water properties or nutrients? There's important nutrients like for phosphorus and nitrogen for biogeochemistry. How are they affected by this process? And then maybe even more provocatively, but potentially more importantly, what are some of the broader impacts? If, again, we want to be able to develop climate models that predict what's going to happen in 10 years or 30 years or 50 years, and we don't have this effect in any of them, how does that impact the fidelity of those model predictions. Could the sequestration of carbon be enhanced by these organisms? Could it be reduced if those organisms are no longer there because of overfishing, because of the effects of harmful algal blooms, other things that we do as we pollute the ocean? These are all questions that came up from this work. And today I'm only going to spend a bit more time talking about this first box, but I want you to keep the second box in the back of your head because these are the questions that we're going to need smart people like all of you to help us think about. So in thinking about what we could do outside the sphere of jellyfish, we know, first of all, the biggest biomass in the ocean is not the jellies, as far as we know. It's things like these krill here. And so they're the most abundant animals in the ocean. They're located in areas that we know are climatically important in terms of places where changes can significantly affect that ocean conveyor belt that I talked about earlier. The Southern Ocean near Antarctica is a good example. They exhibit huge vertical migrations, well over 100 meters on a daily basis. So they're traversing regions of the ocean where the water properties change significantly. And for us, the nice thing is that we wouldn't have to go to Antarctica to study them because we can study analogs of these organisms in more controlled laboratory environments. So this work has been really pioneered by two students in the lab, Monica Martinez, who worked in my lab and finished in 2015 with her PhD, and Isabel Houghton, who soon will get her PhD. The grad students will know how that works. You never are exactly sure. But the work that they're doing is in trying to understand how we can study this problem in the lab. Now, in the laboratory, we're using a different organism. We're not using the Antarctic krill, but something you can buy at a hobby store, Artemia salina or brine shrimp. These are sea monkeys, is another name that they're often known as. Turns out they're very easy to work with in the lab. They exhibit the collective swimming behaviors that I described a moment ago. The other thing that's important is that they have a phototactic response. So here I turn on a laser, and you'll see these organisms run to the laser beam, and then almost like an alien movie or something like that, they get beamed up. Let me play that one more time if I can, because I think it's kind of cool to see. And so what I want to show you is a laboratory experiment that we do in order to see the same phenomenon. There we go. Okay. So in the laboratory, we have a tank. It's about 4 feet tall, and there's a bunch of lasers. So this is like a laser light show in the lab, and I'll try to explain to you as this video plays why we have all of these lasers here. But the main thing to keep in mind from the slide before is that these animals are going to respond to lights in particular ways. So the way that we start this experiment is, first of all, we introduce animals at the top. We turn on this blue laser. The animals will all swim down because they like the laser light. We use this green laser here to center them, and then we make this blue laser rise up, and the animals chase after that blue laser. And so in the laboratory, we can make them do vertical migrations dozens of times a day. They don't seem to get tired. They just go up and down, which is kind of nice. Whereas in the ocean, of course, you get one shot at this every day. They do it at sunrise and sunset. Now, the red laser's here just because we also want to be able to measure the flow. And so like Kakani mentioned in her TED Talk, we're going to use the same type of technique. The red light is less of a stimulant than the blue or the green. So it allows us to see the flow without disturbing the animals in their perpetual up and down swimming cycle here. So here's a still image of the laboratory experiment here. This is a later iteration of it here. You can see there's a double reflection here just because of the edge here. You can see the green light and the blue one. And here you can see the experiment in action. So here's the blue laser rising up on the rail, and then you'll start to see some speckles here. Those are the Artemia chasing after the blue light. And so in the laboratory, again, we can do this to create these on-demand vertical migrations, and then we can study how much mixing these animals create during this process as they're continually making these vertical migrations, as you can see here. And so this is work that we've done over a long period of time, and I'm going to try to wrap up here by just summarizing some of the current findings. And again, the key question here is, do the animals create flow features that are bigger than their individual bodies? They're only a few millimeters each. as they move together collectively, is it possible for them to create larger scale motion? So let's start with the smallest scale. Here you can see a single one of these organisms swimming through the field of view. I won't go into the technique here, but here in Southern California, I lived down here for a good 15 years, so you get these very hot days where if you're sitting at the traffic light or you're sitting in traffic, you can see the heat sort of rising and it creates this sort of scintillating effect. So what you're looking at is the fact that the light rays are being bounced around by differences in the density of the air. The really hot air that's rising has a different index of refraction, as we call it. So light bends as it passes through, and so you get this sort of effect. We can do this in the laboratory where, in this case, it's not the temperature of the different layers that's making that effect, but we just put salt in the water at the bottom, and we have fresh water at the top, or a much less salty layer at the top. And so now when the different regions of density mix, you start to get these really striking effects. And so this is a way that we can visualize flow features just based on the optical properties of the water density. And so in this particular case, the main thing I want to show you here is that as this animal swims, as this video loops, the organism creates this rearward jetting effect in these eddies here. And then because this water here was heavier, it was heavy, it was pushed up here, it starts to re-stratify. Now if I zoom out a little bit, this is a vertical migration like the version I showed you before. The main migration, it kind of looks a little creepy, I guess, with all these things scattering around. I've never seen it this big before. But the primary migration is on the right-hand side. So we have our camera fixed just to the side of it, so we're not just seeing organisms. What you'll see in a moment is that you get this big bolus here of fluid. And again, this eddy here is bigger than any of the individual ones. And so that was our first hint that in this case too, there's some collective effect where each of them might be kicking the water backwards just a little bit, but collectively they're having a larger effect. But this technique that I just described is limited to only a few centimeter field of view. So we wanted to be able to zoom out. And so this video, here's the tank that I showed you, and this gives you a sense of scale. We start to get to a bigger view. And the way that we create this video here is by taking those PIV particles, those small glass beads. We put them in the column, and let's see if I can hopefully get that to play again. I won't mess with the screen. Okay, there was the previous slide. Again, I'll just, since I'm here, remind you of this effect here. Now on the next slide, we have those particle traces. And the thing I wanted you to see is there's this downward jet of flow, and you're starting to see these larger eddying motions. The individual animal size is indicated down here in red. And so the reason why they look longer is because we've overlaid many frames so that your eye can pick up the streaking motion. But the animals themselves are tiny compared to these really large eddying motions. And so that again gave us a hint that we were starting to see these larger effects. And so we zoom out one more time for this next experiment that I'll show you. And so what we did was we put a layer of dye just at the top. We make it much less dense than the rest of the water, so it sits up there and floats. But again, as these animals are swimming upward, they're going to be kicking water downward. And we wanted to see again whether the animals, this is 10 centimeters here now, so one of these animals is going to be sitting in a box about that big. We wanted to see whether the animals could affect that layer, which sits just up here above your field of view. And so this video will play, and it sort of answers the question very quickly. This is played at 10x. You get this huge downward motion and this huge eddying effect of the dye. Again, the individual animals, to give you a sense of how big they are, they're the things that are casting these shadows here. So they're only as big as the thickness of these streaks on the side. But in this tank here, you're getting this huge effect in terms of mixing. And so it turns out that very clearly the answer is yes. These animals can create this mixing effect much larger than their body size. And the other thing that's nice about doing this experiment in a lab is we can then actually measure how much actual mixing happened. We know what the density looked like before the animals started swimming. We know what it looks like after. They mix 1,000 times more than what pure molecular diffusion would occur. So that's the difference between taking 15 minutes to stir your coffee and doing it in 1 second. So it's a huge effect that these animals are having, at least in this laboratory experiment. And so This gives us a lot of confidence to then say, what happens in the real ocean? And this is where I'm going to sort of leave things for you today, sort of starting where, or ending where I started. We just don't know enough about the ocean to be able to pinpoint where this effect would happen. But those questions like which organisms, what locations and seasons, what water properties, nutrients, one of my colleagues pointed out, one of the things that we hadn't thought about until very recently is the diversity of microbial communities in the ocean from the surface to depth, which are hugely important for things like fixing nitrogen, for sequestering carbon, those processes that I talked to you about earlier. Those little microbes, of course, can't get anywhere by themselves, but these animals might be carrying them along for the ride and introducing them to locations that they wouldn't otherwise get to. Again, 1,000 times slower molecular diffusion versus what they're carrying them. And so how that might play and what sort of feedbacks might occur, these are all open questions that I'm excited to present to you because we need all of you in the room to sign up and help us figure this out. And then on top of that, of course, there are those broader impacts. How do we incorporate this into a climate model? An organism that is only a few centimeters in size is impossible to resolve in current climate models, which might have a resolution of a few kilometers at best. So how do we come up with clever ways of incorporating that? If in some cases these are groups of organisms that are being overfished, how do we deal with that challenge? And might that lead to some negative feedback loops where we start to see tipping points in the ocean? These are all questions that need further study, and hopefully I've said enough here to encourage some of you to take up the challenge, and so that in a few years you can be up here and tell us the answer to these questions. Let me finish by just acknowledging a lot of people. The fun part, one of the fun parts of my job is just getting to work with a really great community of scientists, especially as students, and so I told you a bit about these 4 scientists here. Sean Colin and Jack Costello are great collaborators I've had going back probably as long as I've known Mike Schott. We've been working also on these jellies. A whole host of other folks, especially when you're doing field work. You don't just show up in a boat and start doing work. There's a lot that goes into this, and so we've been fortunate to have a lot of support. The font size here doesn't do justice to Mike and to Kirsten. They've really been instrumental in our research in so many different directions, not just the work I've talked about today, but I could tell you about 10 other projects. I guess we'll go to the gift shop, and I can actually do that. And then lastly, funding from a variety of sources. It's interesting to sort of circle back now, because now we have some very helpful funding from a lot of organizations that kind of snickered when we first started talking about this work 10 years ago. But I think people are understanding that it's maybe not so crazy. And if it's not crazy, then we should start finding some answers pretty quickly. So I will leave you with those words of Walter Monk. Work on problems you most enjoy, strange things happen underway. And I'd be glad to take any questions you have. Thank you.
John, thank you so much.
Of course,
That was wonderful. And now's the time that you all get a chance to ask a few questions.
Yes, please.
Remember that we're going to go into the gift shop afterwards.
So I want to ask you about these great volumes of trash that are floating in the ocean. I want to know if they move a great deal, and also if they affect large enough amounts of water in their area that has an effect on the small life that's in that part of the ocean.
Right. So the question was about the large trash, frankly, that's in a lot of the ocean. We know about the large garbage patch and other concentrated regions of plastic and other things that have been disposed of. That's a very active area of current research for a couple of reasons. 2 that are particularly relevant here is that those plastics will tend to degrade to smaller particles. And those smaller particles over time can often sink if they become negatively buoyant. That sinking process is sort of the opposite of what I showed you in those animations here. So in the animations here, we thought about that sphere rising up and carrying water with it. Those particles, as they sink, or in other particulate matter as well, could then lead to vertical transport downward. That is something that we don't currently think about in terms of modeling the impacts of those particulate. Now, the other important area in which that shows up is in the food web. So those small particles, as they break down, get ingested by things like the jellies that we study here. Many other marine invertebrates and vertebrates as well will end up consuming them. And so that becomes a huge issue. In the popular press, the issue of microplastics has come up in the past couple of years because you're sort of directly depositing into the ocean that small particle size. But even those larger structures that you're thinking about in the ocean garbage patch, they break down and eventually will become microplastics that will have similarly negative impacts. So it is a challenge. I think one of the things that would be important to study is where in the ocean those particulates end up. So we know they sink, but again, once things get below a couple of meters below the surface, we don't really know where they end up unless we catch organisms and find them in their guts, for example. So there's a lot that still needs to be studied there. So the question was whether the garbage moves around in the flow or if it stays in a particular area. It's very size dependent. So there's some of my colleagues at Stanford, for example, are looking at this exact question. And so the smaller particles, they're more easily swept away and carried by currents to different locations. The larger particles are more inertial, as we say, and so they'll have much longer residence times in one particular location. So one of the things we need to understand is that degradation process. How does a full milk carton or something like that break down and become small plastic, and in that process, where does it end up? These are open but important questions. Thank you. Yes, sir.
So I saw that you're also a mechanical engineer.
Yes.
And it's about that I'm curious to know if you use tools that can model these environments and use computational fluid dynamics tools and things like CATIA and other things like that. If you, it seems like you're doing a lot of lab work that would preclude that, but I imagine there could be a lot of pressure for you to advance this to the point where you can apply a model or so I'm just curious about that.
Absolutely, and that's a key aspect of the research. So I'll start from ocean-based research, which you might say is closest to the answer of what actually happens, but also is most expensive. It can be $10,000 to $50,000 a day running a research vessel. And so that was what really motivated us to build this laboratory apparatus. But even that laboratory apparatus has a lot of challenges in operating. So it's not something you just flip a switch and the animals swim like they do in the video here, although we cut the videos to make it look like that. So it's hard to get that to work the way you want to. And so ideally, you'd like to be able to simulate these things in a computer, change the model for the swimmer, have that different animal swim, change their spacing, see what happens. We're not there yet, in large part because even with the great computational power we have today, it's still a challenge to bridge the range of length scales we're talking about. So getting from the individual appendages of the organism, which is a few fractions of a millimeter, to tens of meters at which you'd want to be interested in ocean mixing is a scale that even the best supercomputers will take more than a student's PhD thesis to compute. So we're getting toward that with some models. A colleague of mine, Eckhard Mayberg at UC Santa Barbara, has developed some computational tools for simplified versions of the swimmers. And so we're working with him to take our lab data and try to create some of these types of computational models. So it's a work in progress. Thank you. Yeah, sir.
Does the biomass mix rate affect the CO2 absorption of certain areas or not? And can that be affected by things that we can't control?
Right, so the question about was about CO2 absorption and its potential connection to these biomixing effects. That's one of the things that you might be able to get at from the surface in a way that would be interesting to study in the field still. One of the challenges you have is, in that earlier slide, I showed the wind, for example, as an additional effect that's occurring. So you'd want to find locations where you have this animal effect, but you don't have, say, wind forcing. And in many of those places where you might see a signature of change in sequestration, you have both. And so what we would typically then want to do is either, going back to the previous question, do a numerical simulation where we could simulate that process. The challenge there is that air-sea interaction is still a very challenging thing to simulate in a computer. How the sea foam is created as air gets entrained into the ocean, what that process looks like. So that becomes a challenge. The other possibility is finding unique geographic locations where you only have certain of these phenomena. And in fact, one of the reasons we wanted to work in Jellyfish Lake was we had this hope that because, I don't know if any of you have had the chance to be there, but it's pretty well protected. These are old remnants of volcanization. And so you have a protection from wind forcing, and so we thought that might allow us to isolate the effect of the animals. But it turns out that there's lots of lateral fissures, and so you still get tidal influxes. And so you still can't pinpoint and say the animals are the only thing responsible in that context. So it's a very good question. The challenge is finding a controlled way to pinpoint that the animals are the things doing it, absent the experiment which you don't want to do, which is getting rid of the animals and seeing what happens. Yeah, in the back.
Just like not on a big scale, but on a small scale, just because you showed that viscosity they seem to have a bigger impact, do you think it's possible that when you go down into the mesopelagic and the deeper, where it's a little saltier, perhaps the water's a little more dense, so there could be a difference in the impact they have of these deeper layers versus more towards the surface where it's less dense? Is there enough of a difference, you think, in density to create any differences in potential mixing between the deeper and more shallow water?
Right. So in the deep ocean versus the surface, it's true that the density is going to change quite significantly. The viscosity will tend to change less unless there's a large temperature difference. So you're more likely to see a big difference, say, from mid-latitudes to the tropics or something like that in terms of the viscosity of the water, because that's a temperature-dependent thing that is more pronounced than the changes you'll see with depth because of pressure, for example. One thing that I think you would potentially see occurring differently at different depths is the mixing depends on the gradient. So how quickly do the properties like salinity, like oxygen, like nutrient concentration, how those occur with depth. So you will see different effects at different depths because of that. So less because of the animal and their interaction with the water, but and more because of the fact that the gradients, as we call them, will be sharper in certain cases. So one quick example, going back to Jellyfish Lake, if you get down to about 15 meters, you get to this really strongly anoxic region there. So at those layers, this could become a really important phenomena if you're actually bringing oxygen to regions that are naturally anoxic, or vice versa, carrying anoxic water to regions where the other microorganisms are harmed by that. So those dynamics, I think, could be at play. Yes, in the front.
Thank you for that wonderful talk.
No, thanks.
And so I'm familiar with your work and your very productive career.
Thank you.
And I was wondering if you can share with us how you identify problems that you most enjoy, and how do you keep up with all the different things different projects that you have going on?
Good question. On the topic one, I will say, probably the first 5 years of our research, it was very much just curiosity driven. I will say, and this is not just because I'm standing here at Cabrillo, but a lot of the interactions I've had with students around some of the exhibits we have here, conversations with Kirsten, even when we had it over dinner today, sparked another idea. So just talking to other people. I think there's a misnomer that the science is about you and your microscope and coming up with that new, interesting, brilliant idea. But I think our lab, we've found success when we're just having conversations with lots of people and lots of different people. So engineers and mathematicians and biologists and chemists who will all look at this problem from a different perspective and ask a question that we would have never thought to ask. And so I would say that the curiosity is really what drove our initial work. I will say, more recently, we probably have become more attuned to the fact that there are real crises that need to be solved, whether it's climate change, whether it's health, diagnosing, better diagnostics in healthcare. And so if we can satisfy our curiosity, but also come up with tools that can help society, then that's a huge win-win. So we've tried to pair those two together, I think, in our more recent work. Yes, in the back.
We
Those were, you mean in that cartoon sketch? Yeah, those were currents on the ocean.
How does the displacement of density create motion?
Right. So maybe the simplest way to think about this is starting from the ends. So you have water that's being warmed at the equator, and if that water, if the conveyor belt's already in motion and it's traveling toward the poles, it's going to cool because it's going to go to a region that's colder. It's going to get heavier and it's going to sink. And so that sinking motion is going to be really important for driving things down. And so now the question is, okay, how is it that the ocean just doesn't fill up as a pool of cold water? And so you need some energy input then to mix things back up to sort of complete that loop. And that's the initial question that Walter Munk talks about in his abyssal recipes is what are some of the physical processes that enable that water that has just sunk because it was cold and more dense to be mixed back up. And so eventually people have identified that the wind at the surface, almost like, sort of rubbing your hand on a literal conveyor belt, sort of shearing the surface of the water can lead to a driving of the motion. They've also observed that the pull of the moon, the idea that seemed lunatic to people when Walter Munk and Carl Wunsch first proposed it, that leads to a sloshing in the ocean, basically, the differential gravitational pull on the water versus the Earth's crust that leads to those tidal currents. That's another effect. And then potentially this could be another effect that closes that loop. Yeah, in the front.
Say all this research pans out and you conclusively prove that biomass is responsible for a significant portion of swirling of the ocean. How do you apply this knowledge to the way the human race conducts itself? How do you transmit this important information to industry and government in such a way as it will affect positive change?
That is a great question. It's a wonderful question. I will start by, maybe I'll start with the premise, which is, if it's correct. And I think it'll be important to have very concrete evidence that this is something that's important, because even in cases like the fact that greenhouse gases warm the earth, you have concrete evidence, and it's still a challenge to get government action on those types of things. So there's a whole layer on top of if the science is correct that is just, I wish I knew the answer. I will say in this particular case, there are some concrete things that can come to mind, particularly as it relates to how we impact the biomass. So things like overfishing, things like pollution that we contribute to eutrophication or acidification. There are certainly ways to regulate in the same way we were able to deal with the hole in the ozone layer by, banning the use of certain CFCs, for example. There's practices that could be better regulated if we see that they're negatively impacting the biomass. Now, I have colleagues at Stanford and elsewhere who will take a more controversial tack and say, maybe we could enter the field of what's called geoengineering. And so the idea of geoengineering is to say, at a very large scale, if, for example, we know that jellies are effective in sequestering CO2, as we said before, maybe we just put more of them into the ocean and hope that they can actually amplify some of these effects that are positive, or if they're harmful for that process, we remove them. Now, I'm very cautious about anything like that because we simply don't know enough. Going back to the simulation question, we can't possibly simulate what's going to happen if we do that, and so it seems very dangerous to me. But there are people who advocate that as the end game. If we know this is true, then let's modify the organisms because we can manipulate biology in some pretty striking ways these days. But ultimately, I think your question is an important one that touches on policy, how we communicate the science, how the public is galvanized to vote for leadership who cares about these things. And that's the next talk, I guess. Great question. Okay, sure, yeah, absolutely. Let's see, right there. Yeah.
You mentioned marine microbes. Have you spoken to anyone or any marine microbiologists that might be doing similar work to this to understand more of the mixing?
Only very recently. So I was giving a tour to a colleague of mine she's a biogeochemist, and showing her that laboratory experiment. I think her knee-jerk reaction on the principal question was, well, of course they're not mixing the ocean, so why would I care about the interaction between these? But then we start to talk about some of the potential impacts, and to the previous question, if it's correct, what are the impacts? And her perspective is that, well, it's actually a pretty significant thing, because the microbes that are in the ocean soil, for example, at the bottom of the seafloor, those are in many cases entirely different communities from the ones that are midwater or surface. And so if there are mechanisms that are bringing these into contact, then there's a lot of interesting dynamics that just don't get currently studied. I think she and other scientists, I think, are wisely waiting to see whether it pans out, because you don't want to spend a whole line of research answering that question if it turns out that in the end this is a niche effect. Yeah.
John,
Okay, thank you. Oh, wow.
a token of our appreciation.
Thank you very much.
Thank you very
Really
much, John.
appreciate it.
It's great. It's great having you here tonight.
Great to be back.
Good.
Thanks.